So we've got a big, chunk of questions because it's the final. There's a huge number in here. And we're gonna do like 5 in a set. Here's the other thing I realized. Oh, okay, we are recording, good. We'll do like 4 or 5 and then see if we wanna skip ahead. Okay, so this is gonna require you who are here to speak up so I'm not, waiting forever or being the only one to supply the answer. Okay, number 1, the study of biological molecules is referred to as, this should be easy.
Biology.
Torture. Yes, biology. Okay, yeah, that one was good. Is it biology or is it biochemistry? What do you think?
It'd be biology.
Oh, somebody's bringing a balloon over. Okay, so this is where if you're not sure which one to pick, what do you do?
When in doubt, Charlie out. Just pick C and move on.
When in doubt, you can say, so like if you wanted to say biochemistry definition in Google, then that one, the definition says it's the branch of science concerned with the chemical and physiochemical processes and substances that occur within living organisms. And if I do biology definition, then it's the study of living organisms. So which one do you think would better fit? So think molecules is chemistry and biology is living things. Well, molecules and living organisms would more appropriately be biochemistry. Okay, number 2, phylogenetic classification. So that's where things are on branches. When you draw them based on connections in DNA and fossils. So phylogenetic classification differs from taxonomic or group classification, and that phylogenetics is, okay, so do you know which one is based on genetics and which one is based on morphology or physical structure?
Taxonomic would be morphologic, right? Morphologic.
Right, taxonomic is morphology, and phylogenetic is genetic relationships or DNA. So phylogenetics is newer because we didn't know about DNA a few hundred years ago. It's much newer and it's based on genetic relationships. Okay, number 3. The textbook lists 7 characteristics that living things possess, characteristics of life. The process of thermoregulation also is an example of what? So adjusting body temperature through behavior physiology. There's a term here that thermoregulation applies under.
Maintaining homeostasis.
Yeah, so maintaining internal, cellular status that isn't the same all of the time, but has balance, so it doesn't get too far to one extreme or another. You know what happens if a cell reaches equilibrium? This is bad cell humor. It's dead. You don't want equilibrium because that means all the processes stop. But you do want balance. Okay, all the following are features of the scientific method except. So find the one that doesn't fit. Does hypothesis fit?
Classes.
Yeah, there we go. That one was easy.
That was one of those eyesore ones where it's just like that.
And then you go back and you're like, hypothesis, check. Inductive and deductive reasoning, oh yeah, doing theories and making predictions. Formulation of conclusions, that still makes sense. No weird words thrown in there. Observation and experimentation, and you're happy with your answer. All right, number 5, a good scientific hypothesis is all of the following except testable, clear and concise, falsifiable, proven true with data. And I heard someone say the last one. Yeah, here's the bad thing, proven. When somebody says prove it, it's actually not really possible in science. You can get so close to the truth that you believe that it is true, but you never really truly prove it. You just make it more and more impossible to disprove. Okay, number 6. A scientific theory is
generally accepted as true based on data and testing.
Yeah, that's right. So it's not a single tested hypothesis. You have to have a body, a substantial body of data, multiple connected supported hypotheses. So when somebody says, I have a theory in day-to-day life, what they really mean is they have a possible explanation. They don't mean that they have a scientific explanation that was based on, sound evidence from stringent hypothesis testing. So it's a misappropriated word in the world. Okay, number 7, researchers concluded that all organisms are made of cells after examining thousands of different species. What type of reasoning was involved here? So if we go from lots of specific observations to a generalization.
Deduction.
Is it deductive or inductive? Which one is hypothesis testing and which one is theory building?
Theory building would be induction.
Yeah, so if we're getting a theory that all organisms are made of cells, then that is induction, okay? If I say, okay, I've examined thousands of little things inside living organisms that we're calling cells, and every single thing that I've said is a living organism is made of cells, then I'm going to conclude that living things are made of cells. And my generalization, my theory that's been supported is that all organisms are made of cells. So now I can make a prediction. If I find something that's made of cells, then it should be alive. Or if I find something new crawling around and I take a little slice of it and I put it under the microscope, what should I find?
Cells.
Cells. So both of those are predictions, narrow scenarios. Sorry, I don't know what's going on there. Where we've got an expectation, a prediction about what we will find in a specific circumstance where we apply the theory to a single instance. So this bunch of specific to a general category or theory, that's inductive. All right, number 8, a theory as defined in scientific discourse.
An established fact about the natural world.
That would be a law.
Oh, man.
Close. So which on the other one? And the first one, an initial tentative explanation, that's a hypothesis. And a belief, a belief is, gee, that's nice. Something that has to be founded in observation or evidence. A theory, scientific theory, has to be, so when we talk about the theory of gravity, we don't mean that like one day somebody's going to walk out and prove that everything we ever learned about gravity is wrong. And there's actually a theory of gravity, and then there's a law connected to gravity about, what's going to happen if I, drop a brick off the top of a building, right? But we can explain why objects fall based on a whole bunch of evidence about those things happening that explains an aspect of the natural world around us, and in fact explains things far off of the earth, out in the solar system. So there's a theory. All right, this one may look familiar. In 1668, Francesco Redi did a series of experiments on spontaneous generation. He began by putting similar pieces of meat into 8 identical jars. Now think this is back when you would go to the butcher and it's an open air market and you would point to something and say, that slice, please. So, it was common for flies to just hang out on meat. Meat wasn't refrigerated, and what happened to meat? It spoiled, right? And so people thought that the maggots just like appeared in the meat and there was nothing you could do about it. And so he's got these 8 jars, and he leaves 4 jars left open to the air and 4 are sealed, okay? And then he did the experiment again with a 1 variation. Instead of sealing the jars, he covers them with gauze, and the gauze excludes the flies while allowing the meat to be exposed to the air, okay? So he had 4 that were sealed, 4 left open to the air, and then eventually he had 4 that were sealed and 4 that were open to the air but covered with the gauze. And basically, he had each treatment replicated. So this question is, why was it important that he replicated each treatment instead of just having 1 jar of each?
To reduce
the likelihood
result.
of getting an accidental result.
Yeah, no fluke single time that it happened. You know, you would be tempted to choose to practice his technique to make sure the experiment was done correctly. Well, hopefully he knew how to screw a jar lid on before he got started, right? So this would come before. I know that's funny, but when my fish hid under the platforms that I built out in the field and I had to scrap all of my data, I had to practice my technique, but I couldn't use the original data, so it wasn't going to help me very much until I corrected it. All right, number 10. Which of the following is an example of deductive reasoning? So which one sounds like it's a hypothesis or prediction?
Which one sounds like a hypothesis? The 4th one, if a new species of bacteria is discovered.
Yeah, so this is saying we've got a prediction of a specific scenario, so that's deductive reasoning. These other 3 are all generalizations. They're all inductive. Okay, we're gonna skip ahead a bit past the last couple from chapter 1, and let's talk about some things in chapter 2. So oxygen and hydrogen differ in their electronegativity. So you would have to remember what's electronegativity, the tendency of an atom's nucleus to do what? Hang on to its electrons, right? So hydrogen and oxygen, do they share electrons equally? Do they share electrons but unequally? They can't share electrons. Hydrogen's attracted to oxygen but cannot bond with it.
They share them unequally.
That's correct. Okay, good. Do you remember where the electrons spend most of their time when oxygen and hydrogen are covalently bonded together?
In the oxygen.
Going around the nucleus of the oxygen, yeah, because it's bigger and stronger than that of the hydrogen. So that's how you get a polar covalent bond. Number 14, how many electrons are involved in a single covalent bond?
Well, technically 2.
Yeah, 2. 2 electrons are being shared, 1 from each of the atoms involved in the covalent bond. Great. Okay, 15, ions form to do what?
Fill the valence energy level.
Yeah. So when one of those ions becomes the positive ion, the energy level below it becomes its valence shell and it's already full. Number 16, what lacks a charge and where is it located?
Well, neutrons lack a charge.
And they're in the nucleus. So fortunately there was no neutrons being, as another choice. So if you knew neutrons were neutral, you would nail it right off the bat. Okay, good. Number 17, sulfur has 6 electrons in its valence shell. How many covalent bonds would you expect it to form in most circumstances?
6.
So
6 or 3.
you're tempted to say 6, but remember, do you remember how full it's going to be? How many does it need to have a full valence shell? So you might have to grab a periodic table and have it available.
It's like 2 and 6. right?
So it has, so, right, so what's the atomic number of sulfur? It's 8, right?
Mm-hmm.
So what you need to remember here is actually its atomic number is 16. It's in the 3rd row, but whether it's in the 2nd row or the 3rd row of the periodic table, its valence shell is going to have room for 8 electrons. So if sulfur has 6, it has room for 2, and it fills them in pairs. So and already before it's sharing, a couple of those electrons are going to be partnered up, because you could draw it and basically you would see that it had 6 dots around it, and I'm looking up the Lewis dot structure just to plop it up here for you. I'm just throwing up my Google screen here. Can you see this sulfur right here? So we would start and we would go 1, 2, 3, 4, and then we would carry around 5, 6. So now it has 2 spots. Okay, so because it has room for 2 electrons, it's going to form how many bonds? 1, 2.
I see. Okay.
Oops, I just closed way more than I intended to there.
2 more.
Sorry about that. Oh, how did I do that? Give me a second. That's going to be fun. Okay, somehow when I threw it up on top to the other screen, it decided to like take over and not just make a second tab. All right, so it's going to make 2 covalent bonds in most circumstances. Okay, next one, number 18. A molecule that lacks any difference in electrical charge at one end as opposed to the other. So it doesn't have an imbalance of electrical charge. What would we say that molecule is?
Non-polar.
Non-polar, exactly. Great. Number 19, an uncharged atom of boron has an atomic number of 5 and an atomic mass of 11. How many electrons does boron have? So you have to remember what atomic number represents. Atomic number is the number of protons, which in a neutral atom is always the same as the number of protons. And the atomic mass is the number of protons and the average number of neutrons in the nucleus. So how do I figure out how many electrons? It's the same as the number of protons. So it's going to be 5. Number 20, what kind of bond would you expect to form between 2 carbon atoms? So because it's 2 of the same thing, their electronegativity values are equal, so the difference between them is 0. So what will it form?
Non-polar covalent bonds.
Non-polar, excellent. Okay. A bog with a pH of 2 is alkaline. So you have to remember, hey, is pH acidic or basic? And hey, which means alkaline, acidic or basic? And if you don't remember, pH 2 is acidic. And does alkaline the same as acidic, or is it opposite?
Opposite.
It's opposite. So if a pH of 2 is acidic, then is it true that it's alkaline or false?
False.
False. Good. Okay. Why is water a good temperature stabilizer? Oh, it looks like this one, I must have started to type something. and not hit save. So there's no third option. So water is really good at stabilizing temperature, at dissolving substances, and ice floats because solid water has less density than liquid water. These properties of water all have to do with hydrogen bonds, but there are the three answers here refer to hydrogen bonds. What has to happen in order for the temperature of water to rise? We have to break hydrogen bonds first and then be able to heat the water molecules up and get them moving. So the hydrogen bonds keep the molecules from changing how much they're moving, how excited they are. We have to put a bunch of energy into breaking the bonds before we can change the motion, increase the kinetic energy of the water, okay? The hydrogen bonds are temporary, so there's no way they can keep the molecules locked in a permanent crystal structure. The only time we see a quote unquote permanent crystal structure is when water is frozen, and then all 4 hydrogen bonds are formed. But this doesn't refer to ice, so you can't choose that one. So it would be the second option. And hydrogen bonds don't do anything to promote the formation of more covalent bonds. That's just totally false. Okay, number 23. Oxygen and hydrogen atoms differ in their electronegativity. So what does that mean for these 2 atoms? If you look at this third option, if the oxygen pulls electrons away from the hydrogen, that makes sense. But it would that make the oxygen more positive or more negative?
And make it more negative.
Yeah, so this is tempting because the first part is correct, but the second part is false, okay? They share the electrons because remember, it's a covalent bond, but they share them unequally because the electrons are spending more time orbiting the nucleus of oxygen. So this one is correct. And water is a polar molecule specifically because this second option is correct. All right, number 24. The hydrogen atoms of a water molecule are bonded to the oxygen atoms by what type of bond?
Polar covalent.
Polar covalent, yes. And then neighboring water molecules, so the ones that are adjacent or next to each other, are held together by temporary
Hydrogen bonds.
hydrogen bonds. Yes, so that makes this one correct. Okay. All right, number 25. Are the bonds within a water molecule called hydrogen bonds?
No.
No, it's the bonds between a water molecule that are called hydrogen bonds. So this one is false. Okay, what will 2 positively charged substances do?
Repel each other. Still on your island?
Yes, repel each other. Good. Okay, next one, 27. The backwaters of the Amazon River Basin have been measured to have pH levels as low as 3. This is because of all the rich organic compounds dissolved in it. A pH of 3 means that this water contains a concentration of what? So what does a pH of 3 mean? Where think about pH of 1 is as concentrated as you can get, right? And when we go to 2 and we go to 3, so 3 is acidic, right? So we're going to get rid of, it's not that one, because it's not hydroxide ions. And does it mean that it has a huge number like that? Or, and remember it's a logarithmic scale, so you don't want to choose that one. So a pH of 1 means 1 molar, and we're going off, we have to go off by a couple of steps. So what would a pH of 3 mean?
0.001.
Yeah. So it's I'm looking at if I can show you an example. Oh no, this scale is just showing that it has 10,000 times more hydrogen ions than water. So yeah, it's this one right here. Okay. And I'm looking at that. Something's bothering me about that. Just give me 2 seconds here. Well. Okay. All right, let's move on. Which state of water is being represented by the image? So can you see these bonds right here? Look at one water molecule like this one. How many do you see? 1, 2, 3, 4. And can you see that this has a repeating lattice structure? That tells you that this is a crystal. And what form of water has a crystal structure that has 4 hydrogen bonds?
Solid.
Solid, yes. Great. Okay, number 29. If you place bacon grease in water, well, hopefully it's not boiling hot so it doesn't spit in your face. But you put bacon grease in water, it separates into a film on top of the water. Based on this, which is true?
Hydrophobic.
Yeah, so all of these say bacon grease is hydrophilic, so you know they're all wrong. So make sure to check this one. Bacon grease is hydrophobic and molecules of bacon grease are held together by non-polar covalent bonds. Okay, number 30. Some plants can live in bogs in which the pH is about 2. Thus, these plants are able to live in a what kind of environment?
Acidic.
Acidic, yeah. So they must have metabolic processes that are good at buffering, as in keeping the internal conditions of the cells from getting too acidic. Okay, moving on to biomolecules. The myoglobin protein, which carries oxygen to the muscle cells, has only the first 3 levels of protein structure. So we're talking about primary, secondary, and tertiary, but not quaternary. What can you conclude about myoglobin? Oh, there's a typo. It should say it is not pleated or coiled. Okay. All right. So hopefully that gave you a second to recover what you know about protein structure. So at the primary level, that's the sequence of amino acids, the order in which they are bonded together. The secondary structure is when there are hydrogen bonds that form between the R groups to make either alpha helices or beta pleated sheets. And the tertiary structure is when you have additional folding that gives the polypeptide its 3-dimensional shape, okay? And then quaternary is when you have multiple polypeptide subunits that get bonded together. So you can have a protein be complete at either tertiary or quaternary structure, depending on what the final function of that protein is. So which one makes sense to you there?
3.
Yeah, it is made of only one polypeptide chain. All right, 32. What are the parts of a nucleotide?
2 phosphate group, sugar, and nitrogen base.
Good, good. Some of these are easy recognition knowledge ones, and others are like apply that knowledge to a scenario. Which of the following statements about lipids in animal cells is false?
Many lipids function as enzymes.
That's correct. Enzymes are actually what?
Proteins.
Yeah, many proteins function as enzymes would be a correct statement. Good. All right, trans fats are saturated or unsaturated? Trans fats are
Unsaturated.
they're saturated. Wait, so I'm thinking about hydrogenated fats. Yeah, so trans fats are the ones that are the hydrogenated ones. They're saturated and they're not a good idea to eat them. We used to think, oh, they come from plants, so they're healthy. But that's how we get hydrogenated oils. That's what trans fats are. Because we used hydrogen to artificially saturate and turn a liquid oil into a solid fat. And your body actually doesn't digest them the same way. We can't metabolize the trans fats. Okay, 35. If you shake up a bottle of vinegar and oil dressing to mix it, why do you have to do that every time?
Oil's hydrophobic to vinegar.
Yeah, and vinegar is hydrophilic, so they separate. Oil and water don't mix. All right, let's move past a little bit. Okay, there's another one about structure, so you might want to go back and make sure you understand the connection between primary, secondary, tertiary, and quaternary structure in proteins. Let's take a look at 39. It says which functional group is not present. So you have to think about what you see here. So what is this?
That would be the carboxyl group.
It's an OH, so it's just hydroxyl.
Hydroxyl.
Yep. And then, do you see this COOH? That's a carboxyl group. This is an amino group, and this is a ring, so you don't know what that is. So what is missing?
Phosphate.
A phosphate, yeah. You see no P with surrounded by O's, okay? All right, number 40. What is the Golgi complex or Golgi apparatus used for by cells?
Oh man, because I'm gonna get this one wrong.
So you might want to go back and do what with cell structure? Bust out a learning assignment. Remember the one that had all the definitions on it, the matching? Get that one out and go back through it so that you have it kind of more present in the front of your memory when you take the test and available as a quick reference also if you need it, right? So what does the Golgi do? I'll give you a hint. The smooth ER makes lipids.
Isn't it the editing, packaging, and labeling protein?
That is correct, okay?
It's pretty complex. So.
All right, here's another, just you gotta remember what it is and what it does. What are tight junctions? Where do you find them? There's a diagram from a PowerPoint that shows tight junctions and shows desmosomes and shows gap junctions. Those are the three that are in animal cells.
I'm thinking the first one, but I also think the third one as well.
Tight is an animal, and tight makes things waterproof. So you would find them here, okay? Okay, 42, which of the following is false about prokaryotes? So then you have to think, all right, what do I remember about prokaryotes? What do I remember about eukaryotes? Do prokaryotes have DNA? Yes, but it is in a nucleus.
It's
No, it's not.
cytoplasm, right?
Sorry, say that again?
Is it in the cytoplasm?
It's in a region called the nucleoid, but a prokaryote doesn't have a true nucleus. In fact, prokaryote means that's the definition of prokaryote. It means before the nucleus, okay? So this is true. Their DNA is not enclosed in a nucleus. Are they generally larger than eukaryotes? Like the individual cells?
Oh, I don't think so.
Are bacteria prokaryotes?
Yes.
Yes. Are prokaryotes always single-celled?
No.
No.
Yes. Eukaryotes are not always single celled. But a prokaryote, an individual prokaryotic cell, is always smaller than a eukaryotic cell because it doesn't have organelles. Eukaryotic cells, even if we're talking about a single-celled eukaryote, like a protist, it's going to be bigger than a single-celled bacterium because the eukaryotic cell has organelles. So this one is false. Okay. Number 43. What is a cell wall used for in both prokaryotes and plants?
I would say structural support and prevention of lysis.
That's accurate. Okay, 44. Which of the following best describes the flow of information in eukaryotic cells? So if the DNA is in a nucleus, we have to get an expendable copy out, right?
So DNA to RNA.
DNA to RNA to
Protein.
proteins. This one. Okay, number 45. What docks onto the rough ER during the production of proteins? So what binds to the rough ER in order for
Ribosome.
what makes it rough? Yeah, ribosomes. Good.
Oh.
Okay, number 46. You discover a cell that has a cell wall, a plasma or cell membrane, ribosomes, DNA, a nucleus, and small protrusions or soft spikes coming from its body. So is it a eukaryote or prokaryote?
Prokaryote.
Why?
Eukaryote.
Because of the spikes
But this,
and the nucleus.
it can't be a prokaryote because it has a nucleus.
Oh man. Well, I'm
You
here to
can
get
have-
answers wrong so we learn, right?
So a prokaryote can have DNA, can have ribosomes, and a cell membrane and a cell wall. But could you have something that has a cell membrane, a cell wall, and some spikes that come out of it that is just a funny single-celled thing? Yeah, it would have to be a eukaryote because it has a nucleus. Okay. Oh, here's another one about tight junctions. Do you remember what they do?
Waterproof.
They're waterproof. Oh, I gotta fix that
It's
one.
probably that one.
It is that one, but it should say the movement of fluids. And it's totally, ooh, it's not there. So I'm making a note. Okay, 48. Oh, we're gonna skip that. All right, what are the 4 things that make up a plasma membrane? Hopefully you know phospholipids.
Cholesterol, proteins.
And you're like, what is a glycocalyx? So if you don't remember that, then what do you do? Like really quick, because you have time this term?
Google search.
That's right. And so if I do that, that says it's also known as the paracellular matrix. It's a glycoprotein and glycolipid covering that surrounds the cell membranes of some bacteria, epithelia, and other cells. So it's sort of like this extra protein, carbohydrate, or carbohydrate fat slime that they project around themselves. And you've seen that. If I just grab a diagram here, and I just plop this up here, can you see how there are these proteins that have carbohydrate chains embedded? That's what that is. And so there would be, you know, thousands upon thousands of them sticking out of the phospholipid bilayer that would kind of create the extracellular matrix. All right, now let's see if it will let me go back. Maybe. Oh, this time it did what I wanted it to. That's funny. All right, so it would be this one right here, the third one. Okay, the smoothie R, what does it do?
Lipid synthesis.
Lipid synthesis and detoxification. So you'd find more of it in the liver than in other kinds of cells. Okay, so you might need to review cell structure depending on how that went for you. Here's a question for you. What role does cholesterol play in the plasma membrane? I made a Zoom video where I went over some questions about the membrane structure, and I had one with Caribou on it. And basically, cholesterol helps the membrane to either stay fluid in low temperatures or not get too fluid in high temperatures. So basically, it's this one. It helps maintain optimal fluidity. It keeps the lipids together, not too far apart, and also helps them from getting squeezed too close together. Okay. Kids, go back upstairs, please. All right, we got a few more minutes. Should we skip ahead to another chapter that's a little
just skip ahead and get us as much coverage as we can get in.
Okay, all right, here's one. What do you need to pass the M checkpoint?
Complete verification.
What's being checked for in mitosis?
It's the spindle attachment, correct?
Yeah, it's to make sure all the sister chromatids are attached to the microtubules so that we have them separate properly during anaphase. Good. Okay, let's look at 60. Do you remember what oncogenes do?
Vaguely.
Do they help control the cell cycle?
No.
Well, they do help control the cell cycle, because if they're out of whack, then what is the cell cycle? Not controlled.
Uncontrolled cell growth.
Yeah, so oncogenes, think about like proto-oncogene. And what a proto-oncogene does. Basically, thinking about proto-oncogene, a proto-oncogene will regulate programmed cell death. And then I'm thinking about an oncogene maybe actually, so proto would be controlling the cell cycle, but if it's changed to an oncogene, oncologists study cancer, so maybe we like false. So this is where if you're like, ooh, what should I do? And if I look up the definition, then what I get is a gene which in certain circumstances can transform a cell into a tumor cell. So you could ask Google, is an oncogene a functioning gene? And of course, right now I can't spell functioning. And what I get from Wikipedia is an oncogene is a gene that has the potential to cause cancer. And in tumor cells, these genes are often mutated or expressed at high level. So if it's an oncogene or a proto-oncogene and it's functioning properly, then what is it going to do? It's going to control the cell cycle, but if it's mutated, then it's no longer in control, or it's controlling things in a bad way, which could lead to cancer. So we're going to go with true. Number 61, which of the following is not checked for during the G1 checkpoint? What hasn't happened yet?
DNA replication.
Right, because that happens during S. right. Do you remember when cell, oh, so now you get to check yourself. When does cell replication occur?
M phase.
Yeah, good. All right, what's separated in anaphase?
Homologous pairs.
If we're just talking
Or would it be the
about,
sister chromatids?
yeah,
Yeah.
yeah. If it said specific to meiosis 1, then it would be homologous pairs. All right. When does one cell in the cell cycle, so we're just talking somatic cells, when does one cell get divided into 2 cells?
Cytokinesis.
Cytokinesis.
Cytokinesis, yes, because technically mitosis is division of the nucleus, and cytokinesis is when the cytoplasm gets divided, and you have officially 2 cells. All right. So when does the cell carry out the normal functions like protein synthesis? And you would have to choose all that apply.
It's a G1 phase?
G1
Okay.
and
Oh, G2.
G2, yes.
Mm.
All right, 66. The chromosomes become diffuse chromatin, the nuclear membranes reappear, and the mitotic spindle breaks up. When does that happen?
The mitotic. That's telophase.
Telophase, great. All right, one more on the cell cycle and mitosis, and then we'll move ahead for a few more minutes. If a eukaryotic cell has 24 chromosomes and it goes through mitosis, at the end of division, there will be how many cells?
2 cells.
2 cells, and then how many chromosomes?
24.
24, yeah, so we conserve chromosome number in mitosis. Excellent. All right, so meiosis reduces the diploid number of chromosomes by half to prevent the number of chromosomes from doing what?
Increasing.
Increasing, yeah. If we didn't split them in half, then every time an egg and a sperm got together, we'd have double the number of the chromosomes of the previous generation. All right, aneuploidy is always fatal in humans. Do you know what aneuploidy means? Polyploidy means many, so what do you think aneuploidy means?
None.
None? Okay, anu is abnormal. So an abnormal number of chromosomes. It is always fatal or sometimes fatal?
Sometimes.
Sometimes, right? So we're gonna say false. For example, I have a brother with Klinefelters. So he has an extra X chromosome and he's very much alive. And so it's definitely not fatal. Okay, let's see. What is the state of having more than 2 full sets of chromosomes? Do you remember what I just said?
Polyploidy.
Polyploidy, yes, good. Okay.
Poly always means more than or more than normal.
Yeah, yeah.
Like when I was delivering kids, we'd get like polydactyl babies. You'd get them with like six fingers or 6 toes. It's kind of cool.
That is actually a dominant gene.
Really?
And one of my favorite examples. Yes, but it's very rare.
Mm.
So not all rare genes are recessive. Some recessive genes are very common, and one of the best examples is most of the people you'll ever meet in your life are 5-fingered. But yeah, Casey Zimmerman in middle school, she was proud of her scars. A friend of mine. All right, because she had the 6th partial digits removed surgically, or her parents did, I guess.
Oh,
so
man.
a human cell containing 22 autosomes and a Y chromosome.
It's a male.
So if it
Somatic
only has
cell,
22,
Somatic,
autosomes are the same thing. A somatic cell is one that does not make gametes. But here we've got 22 autosomes and a Y chromosome. Notice we don't have an X and we don't have 44 autosomes. So we have a haploid number. So what does that mean we have to have? This is a sperm cell. Yeah, because it's haploid and Y, right? And is it possible That it could be a somatic cell in a female?
No.
No, just the
because there's a
Y.
Y chromosome. That's right. Okay. Do homologous chromosomes split and become daughter chromosomes?
False. That's just my meiosis one.
Yeah, so when we're talking about, well, homologous pairs, yeah, so this is kind of like, ugh, what's going on here? All right. So a human gamete has how many chromosomes?
2.
In, 4 in, or 2 in? A gamete.
Oh, N.
Yeah.
No, 2 N. Oh, yeah.
No,
N.
a gamete is haploid, egg or sperm, and then when the egg and sperm merge together, the zygote has 2 N. Good. All right, let's see. Do you know, 3 of these are ways that meiosis can increase genetic diversity. And so independent assortment is definitely one. And crossing over is definitely one. Non-disjunction is when sister chromatids don't separate. Does that create genetic diversity, or does that create genetic, like, problems? Yeah. And haploid cells that then go on and get used in random fertilization to mix up the genes, that also increases genetic diversity kind of before the fertilization event. So the correct answer here is non-disjunction. Okay. So in which phases of mitosis and meiosis do the same structures split apart?
Jeez.
So when do structures split is metaphase or anaphase? Anaphase, okay. And anaphase of mitosis splits sister chromatids. When do the sister chromatids separate in meiosis? In anaphase 2, the second round. There you go. All right. So the transcription protein, it's not even telling you what it is, but DNA polymerase, which way does it move on the coding strand? When it's, yeah, so you know it's not bidirectionally. So does it move 5 to 3, or does it synthesize 5 to 3?
It synthesizes 5 to 3.
It synthesizes 5 to 3. So what direction is it going to move along the coding strand?
3 to 5.
It's going to move 3 to 5. Okay. And it's going to synthesize 5 to 3. So the new strand that it moves is going to, yeah. All right. 79. What is the name of the protein that synthesizes the leading and the lagging strands?
RNA polymerase.
Leading RNA polymerase is going to make RNA. So
it's DNA polymerase, I think, but I'm not sure which 1, 2, or 3.
Yeah. I'm looking to see if I can give you a place to look for all of them. All right. So, well, I was just checking where I could confirm it here. But it's, so DNA polymerase 1 removes the primers, and DNA polymerase 3 is the one that does both the leading and the lagging strands. So polymerase 1 comes back and removes the primers. So it's DNA polymerase 3. All right, do you remember which end of the DNA nucleotide has the phosphate group? The free blank prime end. It has to be 5 or 3, because remember 5 prime to 3 prime.
It's 3, right?
It's 3. It's a free 3 prime hydroxyl group. Good. All right. So let's do one more here. A biochemist isolates and purifies molecules needed for DNA replication. when she adds some DNA, replication occurs but results in DNA molecules that consist of normal strands paired with numerous segments of DNA. So what was missing? Why do we have a bunch of fragments of DNA?
There's no DNA polymerase.
There's no ligase to seal the fragments together.
Like.
That was my first pick too, and I second guessed myself.
Okay. All right. DNA is what? The two strands go in opposite directions.
Anti-parallel.
Anti-parallel. Good. And do you want to do like two more? Do you want to stick with this DNA stuff or move ahead a little bit?
We can move ahead if anyone else is.
Okay.
Yeah,
Anyone else is good.
Yeah, that's good.
All right. So you might need to, review a couple, have the code on. You don't really need the codon table as long as you know the base pairing. All right. This is getting into chapter 8 right here. And there are a bunch of sample questions that I went over. So skin color is inherited with a single gene that has 4 different alleles. So it's non-Mendelian inheritance, and what's it called?
Multiple alleles.
Multiple alleles, yeah. All right, people with the sickle cell trait are heterozygous for the sickle cell allele. They're usually healthy and have resistance to malaria. So this dual phenotype where they're healthy but have resistance to malaria, and they have both normal and abnormal hemoglobin. So what does it mean when you're a heterozygote and you basically make both proteins?
You're a codominant?
Yeah, because both things are being expressed, codominance. All right, in snapdragons, heterozygotes for one of the genes have pink flowers, where homozygotes have red or white flowers, okay? So that tells you pink has one of each, right? And why is it not still red? Why is it pink? But when plants with red flowers are crossed with plants with white flowers, what proportion of the offspring will have pink?
It'd be 50.
If you're pink, what does it mean? If you're a heterozygote and you have pink, red and white must both be what? Homozygotes. So if I cross a homozygote with a homozygote that are opposite, so like if this is big F, and this is little f, so red is like dominant or whatever it is, and then we have a big F and a little f in the Punnett square in all of the inner boxes, then every single one of the offspring is going to have pink flowers. Because they're all, if I take a true breeding red and cross it with a true breeding white, then all of the offspring have the same genotype, and so they're all going to be pink. But if I were to cross 2 pinks together, then I'd end up with 1/4 pink, 1/4 red, 1/2 pink, and 1/4 white. Okay, 95. When only one allele in a homologous pair of 2 is needed to give a certain phenotype, we say that the allele is blank to the other one.
Recessive?
So the allele that's giving the phenotype is dominant to the other one.
I just didn't understand the question. I just made a guess.
Yeah, that's okay. When only one allele is needed to give a certain phenotype. So if you only need one allele that codes for 6 fingers to have 6 fingers, then the allele coding for 6 fingers is dominant to the allele coding for 5. Most people you will ever meet are homozygous recessive for 5 fingers. They have little f. But if you only need one in a pair, then the one that gives the phenotype we say is dominant to the other one. Okay. Mendel's observation of the segregation of alleles and gamete formation has its basis in what? In cell division. So think about when do we become haploid? When do the parent homologous pairs separate? and that's the connection.
I want to say prophase, but I don't think that's right.
No, in prophase is when the homologous pairs would condense in prophase 1. Metaphase is when they line up, and anaphase is when the homologous pairs separate. And that's why what Mendel observed with certain traits is accurate, because of the way that homologous pairs separate in anaphase 1. And that basically applies further. Remember that those homologous pairs, what the first pair does doesn't dictate what the second pair does. So all 7 of the pea plant traits that he studied, the fact that they obeyed independent assortment means that they were on different chromosomes, the second option. So if this stuff is kind of fuzzy to you, I highly recommend watching those videos that I made, that I pulled up and showed you where they were in the Zoom folder at the beginning before I started recording. And then just to kind of scroll ahead, so you'll have some more things dealing with genetics. And we did I do lots of examples in those videos, okay? And you don't have to be able to do a dihybrid cross, but you have to understand what happens when I take an organism that's a heterozygote for 2 different traits and what it would generate. And so my video talks about first, outer, inner, and last, which is called the FOIL method in math, but it works for the segregation of alleles too. And I'll tell you that it'll be this one here, because we want haploid gametes, but I want you to go watch the video and figure out why, okay? And then there are questions about evolution, about, you know, Darwin, about homologous structures, about the mechanisms of evolution. And so I just recommend that you go back and you watch those couple of videos. And just a reminder here that the exam is open until midnight on Saturday, and you can take it twice, just like the other exams. And I'll pop this open here and just remind you where those videos are. So because it's the end of the term, they're all the way at the bottom. So here's where I go over the word problems, and I start on the evolution part 1, and here are the 2 videos that do part 2. And then for practice with Mendelian genetics and practice questions, it's the second Mendelian one. And if you just want a review of dominant and recessive and what all the definitions are, then watch the first one, okay?